Yeah, for those who joined a little later, I've told the first few who joined that I'll be waiting for maybe a few more minutes before starting so that how many other people will join, and then I can start. Okay. Yeah, someone had raised their hand. Sumit, do you have some doubt as such?
No, ma'am, some glitch in my mobile, ma'am.
All right, no problem. All right, I think these are how many ever people will join for today. I'm hoping. So, welcome everyone. And today we'll be having a problem solving session for week 2 of the Plant Developmental Biology course. All right. So, I'll show you a few questions which are in lieu with the topics that you have studied this week, and they'll all be MCQs, and you can try and answer them, and we'll go over the answers after maybe you guys have tried to see what is the correct answer. Okay, so let's move on to the first question. The question is, which of the following technique can be used to detect organ specific differential expression pattern of a transcript? So option 1 is southern blot analysis, option 2 is western blot analysis, option 3 is northern blot analysis, and option 4 is eastern blot analysis. So what do you think the correct answer is? You can just write down the answer in the chat and I'll go through them. Then we'll see how many of you think what is the answer. All right, someone is saying 3 is the answer. Does anyone think it's something else? Or should we lock number 3? All right. So when you see the question, you have to note down a few things, right? So what they're asking you is for organ specific, right? Differential expression. That means in different organs there will be different expression of a transcript. So transcript is the mRNA construct that is formed from the DNA, right? So that is what different organs and mRNA, right? These two are the things that you have to note down from the question to figure out what the answer is in this case. So, what is you also have to know these options. One is what is southern blot, western blot, northern blot, and eastern blot, right? So, let's see what they are, and then we'll come back to this question again. So, this is the cell, right? And it will have its own protein, RNA, and DNA. So, when you run a gel of DNA, right, an agarose gel, and you perform a blotting of it, that is transferring it to membrane and then labeling it to check for specific DNA, that is called southern blot, right? And the name southern came because of the person who invented this technique. His surname was southern, so that is where the name also comes from. When you do the same thing with RNA, so you are isolating RNA and then running it on agarose and transferring it to a membrane and then detecting it using a probe, it's called northern blot. The person who discovered this was not named northern, just they played on the original name of southern blot. Okay, and when you do the same thing for protein, you run a protein on a acrylamide gel, okay, because protein cannot be run on a agarose gel. You transfer it to a membrane and then you use a probe to detect it, that is called western blot. Okay, and eastern blot is another name for you to detect post translational modification. So it's similar to a western blot, but instead of just finding the protein, you're finding the post translational modifications in a protein. Okay, so in this case what they had asked you for was organ specific differential expression pattern of a transcript, which is RNA. So as you can see, RNA would be this northern blot, right? So the correct answer is in fact answer 3, northern blot analysis. In case you have any doubts, just stop me or write down in the chat and I'll answer them also. Okay. So the correct answer is northern blot. Southern blot is for DNA, which is not what, because the DNA will be the same in all the cells that you take, right? So if you have a cell from a liver, a cell from a heart, right? And you check with the DNA analysis if a certain gene is present, it will be present in both of them because they're from the same individual, and the gene does not, or the DNA does not change from cell to cell in an individual itself, right? Western blot is for protein. Okay, so since they've asked you for the transcript, that is not the correct answer, even though Western blot can help you figure out organ specific differential expression pattern. Okay. So if you do that for the protein, you can figure out what organ is producing which protein at what time, right? So that could have been the correct answer if they had not mentioned the transcript part. Okay. Northern blot analysis is for RNA, and that is why this is correct. And Eastern blot analysis is for post-translational modification, which is again not correct in this case. Okay? So I hope you guys understood this. So let's move on to the next question. Um, in case of any doubts, just please feel free to stop me, okay? Or if you want me to repeat something so that it's easier for you to understand, please stop me, okay? So the question is, you have to match the following techniques with the analysis that can be done with it. So in the first column you have microarrays, RNA in situ hybridization, chip assay, and translational fusion construct. And on in the other column you have subcellular localization of a protein, spatio-temporal gene expression pattern, global gene expression analysis, and DNA protein interaction. So you have to match the column something from the column 1 to what is the use of this in the column 2. Okay. So maybe go over this for a minute and then see amongst these four which is the correct answer. And just write down whatever you think the answer is in the chat. All right. So does anyone think it's something else apart from option 2? I'll give you maybe one more minute and just go over this question and see what you think the answer is, and just please write it down in the chat so that I know exactly how clear this is to you. All right. So I'm going to saying option 1 is correct. Anyone else thinks it's something else? All right. So option 1 is in fact correct. So option 1 states that microarrays are used for global gene expression analysis, RNA in situ hybridization, for spatiotemporal gene expression pattern, option 3 that is chip assay for DNA protein interaction, and option 4 translational fusion construct for subcellular localization. of a protein. Okay, so let's see how that is by maybe going over what each of these techniques is. Okay, so the first one is microarrays. So this is how a microarray works. So imagine you have healthy cells, okay, and you have pathological cells, okay, that is disease cells, or maybe you can take the example of cancer cells, okay. So this is healthy, maybe liver cells, and you have taken cancerous liver cells, okay, and what you do is you will grow the cells in culture, okay, you will isolate the RNA and you will attach fluorescent probes to them, okay, and then you will do hybridization on microarray. So this is a microarray, and each circle that you see will have a oligo, uh, peptide, oligonucleotide, that is small fragments of a nucleic acid for a certain sequence. In this case, uh, this one well will have the oligo DNA or RNA for gene 1, okay. Similarly, each array will have one for different genes, okay. So you have labeled the RNA or from the healthy cells with GFP, and the one from pathological cell with RFP, okay. So for gene 1, okay, this is gene 1, you're seeing that it is black in color, right? So that means it doesn't have GFP or RFP. That means this gene is not active in the healthy liver cell and the cancerous liver cell. Okay. Then you see this well where it is red. So that means this gene is active in the cancerous cell, okay, but not active in the healthy cell. You see this green cell, okay, green well, sorry, and you see that only green is showing. In that case, this gene is active in the healthy cell and not active in the cancerous cell, and anywhere you see this yellow is where a mixture of these two are there, okay. That means this gene is active in the healthy cell as well as the pathological cell. So how this helps you is it will help you figure out the different gene or the global gene expression in different cells, okay. So you can figure out which gene is responsible for this pathological state, okay. The next one was RNA in situ hybridization. Okay. So in this case, what you're doing is you're probing the RNA. Okay. With any kind of probe. Just a second. Yeah. So you're probing the RNA. Okay. All right. A few more people have joined. For those who have joined, I after. Okay. Yeah. So for those who have joined, I will repeat the first few questions that I've taken after I am done with the entire set. Okay. So where was I? Uh, yeah, RNA in situ hybridization, right? So within the cell itself, okay, that is what in situ means in this case. You are going to attach probes to the RNA. That is anything that is going to help you identify or visualize where the RNA is present in the cell. Okay, so this is a Drosophila embryo. Okay, at different stages of development. Okay, and what you can see is you have probed the gene called hunchback. So you're seeing the movement wherever you see this dark purple is where the RNA is. So as the embryo grows, you're seeing in which cells or which part of the embryo, okay, this RNA is active or this gene is active. Okay, so that is what RNA in situ hybridization means. The next one was chip assay, right? So that is chromatin immunoprecipitation assay. So in this, you have DNA protein cross linking. Okay, you lyse the cell, you sonicate it so that you get small fragments of DNA still attached with the protein. Okay, you remove the protein, so you're left with only the DNA, and you analyze it. So this will help you figure out how much or what portions of the genome is interacting with the uh, protein. In this case, it is histone, that is being, you're seeing which part of the DNA is tightly bound to the histone and which is free. So whichever is tightly bound to the histone, that is not being expressed currently, right? Because it is heterochromatin, right? And whatever is free of the histone is euchromatin, and that is currently being expressed. Okay. And the last one was translation fusion construct, right? So this is translational fusion construct, this portion. Okay. So you have a gene of interest to which you've directly attached the reporter gene, which is in this case GFP. So you can see if the gene of interest is being translated to a protein, because your protein will be attached to this GFP, okay, and that will fluoresce, and you can visualize it in the cell itself, okay. So let's go back to the question. So microarrays. So the answer is global gene expression. So you are trying to figure out which and all genes are being expressed in two different kinds of cells. Okay. RNA in situ hybridization, spatio-temporal gene expression. Spatio-temporal means within the space, that is within the cell, and temporal means at different times. Okay, so this will help you figure out both gene expression patterns. CHIP assay was the chromatin immunoprecipitation, that is this DNA protein interaction, and the last one was translation fusion construct, which is subcellular localization of a protein. So once you have that GFP, wherever in the cell you find this GFP, you can understand that your protein of interest is localized there usually. Okay, so that is the correct answer. I hope you don't have any doubt in this. If you do, just write it in the chat. Otherwise, I'll move on to the next question. All right. So the question is, which of the following enzyme is commonly used to partially digest the tissue for better probe penetration in RNA in situ hybridization? You're given four options: cellulase, proteinase, K, pectinase, and none of these. So what do you think the correct answer is? All right, there is one in the chat saying option 2 is correct. Anyone else thinks it's a different answer? Alright, I'm not seeing anything in the chat itself, so I'm assuming everyone either thinks option 2 is correct, or you don't know the answer for this. Alright, so no problem with either. So what they're asking you is, which is the enzyme that is commonly used for partially digesting the tissue for better probe penetration in RNA in situ hybridization. So like we discussed before, RNA in situ hybridization where you send RNA probes inside the cell itself. Okay? You don't extract the RNA, you send the probe inside the cell so you can see where the RNA is, right? So in that case you need the cell to be permeable to this RNA, right? So what these three enzymes do: cellulase, as you can guess from the name, it will break down cellulose. Pectinase, again from the name, it will break down pectin, and proteinase will break down certain proteins. Okay, so from these, which is the correct answer is, it is in fact option 2, that is proteinase K. So proteinase K, what it will do is it will, in the cell membrane, there will be proteins, right? That will be degraded by this proteinase K, and it that will help make pores in the cell wall or the cell membrane of the tissue that you are going to um, use in the RNA in situ hybridization. Okay. So the correct answer is proteinase K. Before we move on, I just wanted to check with you. Are you able to answer these questions even if you're not writing in the chat? Are you able to answer these questions after watching the videos that are online for the week? Or are you finding it difficult? You can just write down whatever you think is happening in the chat. Again, I'll just read them. Are you finding these questions relatively easy to answer after watching the videos from week 2 or week 1? No answer? All right, then let's just move on to the next question. All right. So, um, for a little bit more detail about this, so treatment with a non-specific protease such as proteinase K. So there can be specific proteases which will break down only certain proteins, right? But protease K is a non-specific protease. It will permeabilize the tissue after fixation and can also release target nucleic acid molecules from bound proteins. So in case they are bound to histones or any other protein, because of which if it is bound to a protein already, your probe cannot go and bind to it. Okay, so like RNA binding proteins, as they're saying. So if your RNA has a protein attached to it, and this is the area that the probe also has to attach, the probe will not be able to attach, right? So in this case, this proteinase K will also remove this protein so that you have a free RNA molecule to which your probe can attach. Okay, and making them more accessible for hybridization. So now that we have done this, let's move on to the next question. And the question is, which of the following statements is false regarding a transcriptional fusion construct? Okay, so there are four options given to you again. This is a slight trick question, I think. So option one is we get to know the tissue where the promoter of the gene is getting activated. Option two is it consists of a promoter or upstream region of a gene along with the reporter gene. Option three is we get information about subcellular localization of the protein. And option four is all the statements are true. So, what do you think the answer is from these? Alright, Manoj is saying option A is correct. Option C, okay. Anything else that anyone thinks is the correct answer in this case? Alright, so what is a transcriptional fusion construct? Like we had discussed a little bit before. So it is a fusion construct for your transcriptional product. That is, the mRNA, right? So you're attaching a probe to it. This is your probe, right? So a transcriptional fusion construct this is. So we get to know the tissue where the promoter of the gene is getting activated. That is true. So if you know where the this okay this is the DNA, this is the gene of interest, and this is the promoter. Okay, so if you know that this is the promoter of the gene, okay, you can attach a transcriptional fusion construct, right? So you end up with this sort of molecule, okay, and you can figure out where, like which cell in the tissue, like if this is a brain, okay, bad drawing of brain, but you can see where certain proteins are active, okay. The second one is it consists of a promoter or upstream region of a gene along with a reporter gene. That is also true. So it has the promoter of the gene that you are looking for, and with that promoter, you're attaching a reporter gene, right? And if in the DNA or in the cell, this promoter is active, that means your gene of interest is active. That means this extra piece of DNA that you have put with the promoter, same promoter, and your reporter gene will also be active. Okay, so you can draw parallels from it. And the third is we get information about subcellular localization of a protein. Since this is a transcriptional fusion construct and it deals with mRNA, you don't necessarily know about the subcellular localization of a protein. Okay, the protein could be formed anywhere and it could be transported to any other place too. Okay, what if it's a cell membrane protein? It is formed within the cell, so that means you will find mRNA within the cell, but its end location is the cell membrane itself, right? So this is not true. So technically, what is false regarding a transcriptional fusion construct is option 3, but you can also say that all the statements are true is also false, right? Because there's a not true statement. Okay. But in this case, if you're asked a question like this, it is better for you to answer as 3 than as 4. Okay. Because that is the closest answer to the question that is asked. Okay. So if there are no doubts, I'll move on to the next question. The question is which of the following techniques can be used to study protein-protein interactions? So your option is yeast 2 hybrid assay, a chip assay, a yeast 1 hybrid assay, and a RNA in situ hybridization. So just write down what you think the correct answer is in the chat. All right. One, two, anything else? Any other option that you think is true? All right. Let's see. So I'll tell you the answer first and then we'll go over the technique itself. All right. So the answer is yeast 2 hybrid assay. Okay. Chip assay and yeast 1 hybrid assay are for DNA protein interaction, and RNA in situ hybridization is to figure out where in the cell a certain type of RNA is being produced, right, or is situated. So that is just for RNA itself, RNA localization. So what they're asking you is for protein-protein interaction. So the answer in this case is yeast 2 hybrid assay. So how the yeast 2 hybrid assay works is like this. Okay. So this is the uh, you know uh, diagram showing this. So what you have here is a DNA binding uh, domain, and this AD that you see is the activation domain. So what happens is the DNA binding domain of uh, you know a protein or a transcription factor in this case binds to a certain part of the DNA upstream of the gene of interest. Okay, so if this is a transcription factor for this gene, this particular gene, it will have a DNA binding domain and a activation domain. So the DNA binding domain will attach to this area which is upstream of the protein, and the AD domain, that is the activation domain, activates the expression of this gene. Okay, so AD and DB, that is, like I told you, the DNA binding domain and the activation domain are different functionally and structurally. Okay, so they have different functions as well as different structures. So they can be separated. Okay, and they can be fused to two separate proteins. So you have protein A, which is attached to the DB, and you have protein B, which is attached to the AD. Okay, so the one with the AD becomes the prey, and one with the DB becomes the bait. Okay, so what happens is the DNA binding domain and the bait protein will attach to the area upstream of the protein. Okay, and the if the protein, that is the prey in this case, attached to the activation domain, okay, is not interacting with the bait, this construct will not go and attach to the reporter gene, right? So you can see that it is not interacting, these two proteins, the bait and the prey protein, are not interacting with each other. But in case the bait and the prey protein, like you see here, interact with each other, that means this construct will be whole. Okay, so DNA binding and the activation will happen, and this reporter gene will be expressed. Okay, so in that way you can figure out if two proteins, the bait and the prey protein, interact with each other or not. Chip assay we had discussed a little bit earlier. RNA in situ hybridization we had discussed a little bit earlier, and the last one was yeast 1 hybrid assay. So in this case, the yeast 1 hybrid assay, like, it is different from the yeast 2 hybrid assay because the yeast 2 hybrid assay is for protein protein interactions, while the yeast 1 hybrid assay is for protein DNA interaction. Okay, so the query protein, whatever you're trying to figure out, is fused with the AD domain. Okay, and it is expressed in yeast strains harboring various DNA sequencing upstream of the reporter gene. Okay, so if the query protein binds to a particular target sequence, then you can figure out, like in this case, that the DNA and the so this bait has the DNA sequence with it, right? So in this case, the prey protein is what you're trying to figure out if it interacts with that particular DNA sequence. Okay, so this is for DNA protein interactions. Okay, so I hope you understood that and there are no doubts. Just stop me or write down in the chat if you have doubts with what I'm telling you. Okay, or it's unclear how I've explained, so that I can explain it maybe better. So right now I'm not seeing anything in the chat, so I'll move on. But if there are any doubts, just please feel free. I can come back to this and explain once more. So the question is, Cas9 is a exonuclease, a endonuclease, a peroxidase, and a ribozyme. So let's see what the majority of the people are saying the correct answer is. So 2 is the answer. Do you guys know what the other enzymes do? Just maybe a little idea. Alright, so just for your information, the answer is in fact correct that you guys were saying. Answer is 2. It is the endonuclease, that is, it cuts uh, the nucleic acid within. Okay, so its function is within the strand of nucleic acid. An exonuclease will work on the end. A peroxidase is the enzyme that breaks down hydrogen peroxide, and a ribozyme is a um, you know um, uh, RNA molecule that has enzymatic activity. Okay, so the correct answer in this case is endonuclease. Like you can see here, this is Cas9. It is an endonuclease in the sense that this guide RNA, once it is, it comes in contact with the Cas9, and this particular sequence of complementarity is there. As soon as it figures out, it will cut the genomic DNA at that point. Okay, so it is an endonuclease because it is cutting within the DNA itself, okay, not the end. And it is specifically cutting because of the presence of this guide RNA. Okay, so let's move on to the next question. The question is, which of the following is an example of targeted suppression of gene expression? Option 1 is gamma ray, option 2 is RNAi, option 3 is tDNA insertion, and option 4 is EMS. So, what do you think the correct answer is? Alright, someone is saying it's option 2. Anyone thinks it's something else? So, what they're asking you for in this question is the targeted suppression of gene expression. So, you're trying to suppress the expression of a particular gene, right? So, let's see what gamma rays do. So, gamma rays are cause they cause mutations in the cell. Okay, so what they do is, if this is a DNA, okay, they cause double-stranded break. So what happens is, the repair mechanism of the cell itself is going to repair this, but it is not necessary that the repair happens in the way that it previously was. So whatever sequence was here before the break, it changes slightly. And if this is a, you know important gene or an important part of a gene, okay, it can mess with your cell itself. Okay. tDNA insertion. tDNA is the transfer DNA that is found in agrobacterium, right? It is used to tDNA insertion is used in you know plant biotechnology to add your gene of interest into the genomic DNA of the plant. So it is not a suppression of gene expression. Okay, and EMS is a chemical mutagen that changes the basis. Okay, and causes mutations again. So the correct answer in this case is RNAi, that is RNA interference. Okay, so how that works is, so as you can read here, a long double-stranded RNA, okay, that is dsRNA, is processed in the nucleus by DROSHA, that is RNase enzyme. Okay. It will result in short double-stranded RNA fragment that is exported outside to the cytoplasm. Okay, and that will bind to the dicer, that is RNase enzyme, and cleaved into even smaller fragments called small interfering RNA. Okay, or microRNA. So here you form small interfering RNAs. Okay, then the small interfering RNAs or the microRNAs will be recruited by the RISC complex. That is what is happening here. Okay, and the double-stranded RNA will be changed into a single-stranded RNA molecule. Okay, which is open now to the attachment of your mRNA to it. So the risk with the siRNA or miRNA binds to the complementary part of the mRNA, that is the messenger RNA. And once this binding happens, this mRNA, that is messenger RNA, will be broken down into fragments and the protein cannot be formed. Okay? So in this case, since the mRNA itself is being cut down, okay, since the mRNA is being cut down, the resultant protein will not be formed. So you are targeting the suppression of the expression of a particular gene in that case. Okay, so the answer in that case was in fact RNA. Okay. So if there are no doubts, let's move on to the next question. And the question is, in RNA sequencing, dash are fragments of known sequence that are ligated to the synthesized cDNA. Okay. cDNA is the complementary DNA, and there are four options given to you. Option 1 is adapters, option 2 is random primers, option 3 is oligo DTs, okay, that is deoxy T's, and option 4 is none of these. So, what do you think the correct answer is? Option 1, yes. Alright. So, majority of people are saying it's option 1. Yes. So, option 1 is in fact the correct answer. So, adapters are double stranded DNA molecules, okay, which have a restriction site within them. Okay, and this will either they have restriction sites or they are, you know, normally they have sticky ends. They could be of either types. Okay, so in the case that they have a restriction site within them, they can you can use a restriction enzyme to break down the adapter sequence to give you this sticky end. Okay, random primers are short oligo DNA molecules, okay, that are single stranded and will attach to complementary part of a denatured DNA fragment. Okay, oligo DTs are TTT, that is a DNA fragment which is consisting of only repeats of T. Okay, so in RNA sequencing, this is the flow chart of RNA sequencing. So you isolate the RNA from the sample. Okay, you fragment the RNA into short fragments because there is certain length that can be sequenced. Okay, so you have to bring all the samples to that length. So the best way to do that is to fragment them so that they become small, and then you can see, and it will be a random fragmenting. Okay, so that you have overlapping sequences which you can finally, you know make a complete RNA fragment from. Okay, and you convert the RNA fragments into cDNA. Okay, using reverse transcriptase, you can convert the RNA to DNA, right? And you ligate sequencing adapters. So to figure out which side, since you don't know from your cDNA itself which is the right side, which is the left side, right? So what you do is you attach different adapters to each side, and that will help you in the sequencing. And then you perform the sequencing and you use the data to figure out the sequence of the RNA. Okay, so what you use is adapters because these are short double-stranded DNA fragments of known sequence. Okay, so they can be used for the NGS. All right, so that is the correct answer. So let's move on to the next question. So in order to discover specific cell type markers, the scientists generated a collection of transgenic lines with random insertions of a promoterless reporter gene and screened for a specific reporter activity in the domain of interest. What is this technique called? There are four options again. One is gene knockdown, gene trapping, promoter trapping, and enhancer trapping. So, what do you think the correct answer is? All right, someone is saying option four, option three. Okay. Anyone else thinks it's a different answer? All right. So the majority is three, and the answer is in fact three. It is promoter trapping. So I'll explain the difference between gene trapping, promoter trapping, and enhancer trapping. Okay. But let's figure out it's not gene knockdown. Okay. So in gene knockdown, what you're doing is you're removing the effect of that gene. You're making that gene not work, right? So the question is stating that in order to discover specific cell type marker, the scientists generated a collection of transgenic lines with random insertion of a promoterless reporter gene. Okay, so and what they're trying to figure out is to discover specific cell type marker. Okay, and screen for specific reporter activity in the domain of interest. So the answer is in fact promoter trapping. Okay, because first of all they're using a promoterless um, reporter gene. Okay, so that itself should technically clue you in, but there is another form which uses a promoter less technique. So let's see what that is. Okay, so this is the gene that you're trying to study. Okay, so a promoter trap is when you add a reporter gene which doesn't have its own promoter. Okay, and you hope and you since you're adding it randomly, you hope that it is um, present inside it is put inside the exon. Okay, that is the part of a gene that is going to be you know uh, made into a uh, that will be a part of the mRNA itself, which will be made into the protein. Okay, so in this case you will figure out if this promoter is working or not for this itself. All right. uh, So what was the question? Yeah. So they're trying to figure out the specific cell type marker. Okay. So they're trying to figure out which promoter is being used in this case. Enhancer trap is when your reporter gene doesn't have a enhancer. Okay. And it is trying to piggyback on the enhancer of a different gene. Okay. And you're trying to figure out how much of this gene or this promoter is enhanced by this enhancer gene. So whatever is the amount of reporter gene that is produced will be also equal to the amount of your gene of interest. Okay. So in this case, in the promoter trap, your transcript will have the reporter gene. Okay. And a gene trap, in this gene trap, it doesn't have to be within the exon. Okay. So all you want is for it to be within the gene itself. It could be in an intron itself. It doesn't have to be translated at all. Okay. So that is a gene trap. So you're just trying to figure out if that gene is being transcripted and translated or not. It doesn't have anything else to do. In this promoter trap, you're trying to figure out uh, if the gene is being translated because you want it to be within the exon, that is the coding sequence. Okay. Sorry, not the coding sequence. That is the part of the DNA that will become a protein. Okay. So you're trying to figure out if the protein is formed or not. Enhancer trap is when you're trying to figure out how much it is being enhanced using, or if it is being enhanced using an enhancer. And the gene trap is just to figure out if that gene is being transcribed or not. Okay, transcribed. Yeah. So since it is in the intron, it doesn't need to be translated. So you're just trying to figure out if it is being transcribed or not. So the correct answer in this case would be promoter trapping. And that is it for the question. So for those who joined late, I'll take the first few one again. Okay. Um, those who were there for the entire thing from the start to the end, you guys can leave the meeting. So this is all the questions for today, unless you have any doubts. So I'll give you a few minutes. You can write down in the chat if you have any doubts with regards to some question that we've taken today that we can discuss. So the next time we meet, I think we can discuss the first assignment. I think the due date is 7, right? So maybe we can discuss the first assignment and see where we've gotten correct answers and where we've gotten wrong answers in case that is needed. So are there any doubts regarding the questions that I've taken today? All right, I'm not seeing anything in the chat itself. So I'm assuming you don't. Those who have been here since the start don't have a problem with the questions or any doubts with the questions that were taken today. So you guys can leave the meeting. Those who were there for the entire thing from the question number one, which is, let me go back. Yeah. So this was the first question. If you were there since this question, then you have seen the entire set of questions that I've taken today. You guys can leave the meeting. And those who are remaining and those who joined late, I will do these questions once more. Okay. So I'll just wait for those people who were there for the entire thing leave, and then we can restart again. Yeah, I'll explain translational gene construct, not a problem. So I think I have a diagram somewhere of translational gene construct. Yeah. So this is a translational gene construct and a transcriptional gene construct. The diagram showing that. Okay. So the first type, that is transcriptional gene construct or a transcriptional gene fusion, is for you to figure out whether your gene of interest is being transcribed or not, okay, or it's being expressed or not. So in this case, like you can see, this is your gene of interest, okay, and it has a promoter site, okay, and this is your reporter gene. So the reporter gene will also have a ribozyme binding site, and your gene of interest will also have a ribozyme binding site. Okay. So if this gene is being expressed, okay, and that means this promoter, since the expression of this gene depends on this promoter, right? So if this gene is being expressed, that means this promoter is active, okay, and since this reporter gene doesn't have its own promoter, it is going to depend on the promoter of your gene of interest. Okay, because you're adding your reporter gene just directly after your gene of interest. So if this promoter is active, that means this entire set from your gene of interest to your reporter gene will be transcribed. Okay, and when it is getting translated, since they each have their own ribozyme binding site, they will be translated as two different proteins. Okay, this you won't be able to quantify, okay, unless you probe them. That is why you're using this. So from the amount of GFP that is there in the cell, that will be equal to the amount of your gene of interest. Okay, so that will help if your mRNA that is being formed, is being translated, you will have a GFP protein being formed. If it is being formed but not translated, then you will not have a GFP being formed. Okay, so that way you can use a transcriptional gene construct, and the next is a translational gene fusion, right? So in this case, this is your promoter, this is your ribozyme binding site, your gene of interest, and your reporter gene. Your reporter gene doesn't have its own uh, ribozyme binding site. Okay, so it is depending on the ribozyme binding site of your gene of interest. Okay, so in the first case, you will have a functional gene of interest, and in the second case, you may have a non-functional gene of interest depending on the effect of this GFP being attached to your gene of interest. Okay, so since there is only one ribozymal binding site, you will have a mRNA being formed. Okay, and since that mRNA will have only one ribozyme binding site, your protein that is being formed will be a fusion of your gene of interest and your GFP. Okay, so this will tell you the direct amount of your gene of interest that is being formed. Okay, and your gene of interest will be attached to the GFP together. Okay, they won't form different proteins. It will be one big protein which will have which will have your gene of interest attached to your GFP. Was that clear, Pooja? All right. Any other doubts you have? I'll explain that too, and then we can go on to repeating the question. Anyone else has any doubts? Yes, ma'am, as in you have a doubt? Alright, so it's no. So, since I'm not seeing anyone else having a doubt, we'll just maybe go over the questions that were taken. Alright, so this is the first question. Which of the following techniques can be used to detect organ-specific differential expression pattern of a transcript? In this case, transcript obviously means that it is a RNA that you're looking for, right? And they're asking you for organ-specific differential expression. So, you need to know the difference between these kinds of blots. So a southern blot is where you're trying to find the DNA, a northern blot is where you're trying to find the RNA, and a western blot is where you're trying to find the protein. Okay, and the eastern blot is where you're trying to detect post-translational modifications on a protein itself. Okay, so the question is asking you to find out the expression pattern of a transcript that is RNA. So the correct answer is the northern blot, which is the blotting technique used for figuring out the RNA of interest. Okay, so the next question is you have to match the following techniques with the analysis that can be done with it. Option 1 is microarrays, 2 is RNA in situ hybridization, 3 is chip assay, 4 is translational fusion construct, and what it can be used to do is subcellular localization of a protein, spatio-temporal gene expression pattern, global gene expression analysis, and DNA protein interaction. So the correct answer is option 1. That means microarrays can be used for global gene expression analysis. So global gene expression means all the genes that are being actively translated or transcribed, okay, in different sorts of cells. So if you have normal cells and cancer cells, using microarrays you can figure out what is the difference between the genes that are being transcribed or translated in both of them. So you can figure out what is the genes responsible for the condition where the normal cells are turning into cancer cells. Okay. Then RNA in situ hybridization is to figure out the location or the time at which certain RNA molecules are expressed in a cell. Okay. So that means it is spatiotemporal, that means space and time gene expression pattern. Chip assay or the chromatin immunoprecipitation assay is for DNA protein interaction, or in this case DNA histone interaction, and translational fusion construct is for subcellular localization of a protein. Okay, so if you're doing translational fusion construct, your gene of interest is attached to your GFP or any other probe. Okay, and using the fluorescence you can figure out where within the cell the protein is present. Okay? So subcellular localization of a protein can be figured out using translation fusion construct. So let's move on to the next question. That is, which of the following enzyme is commonly used to partially digest the tissue for better probe penetration in RNA in situ hybridization? So RNA in situ hybridization is when you use a probe to detect the RNA or the spatiotemporal expression of RNA inside a cell directly, okay, instead of isolating the RNA and then figuring out where it is present. So cellulase is an enzyme that will break down cellulose. Pectinase is an enzyme that will break down pectin. The correct answer in this case is proteinase K. Okay. So, treatment with the non-specific proteinase such as proteinase K will permeabilize the tissue after fixation. Okay, and it can also release the targeted nucleic acid molecule from bound protein. So, if your gene of interest is bound to the histone, okay, the probe will not get attached to it. So, your proteinase K will also help remove the RNA or uh, the RNA from RNA binding protein, okay, and they can attach to your probe of interest easily, which will not be possible if the protein is still bound to your RNA molecule. Okay, so the correct answer was proteinase K. Let's move on to the next question. The question is, which of the following statements is false regarding the transcription fusion construct? Option 1 is we get to know the tissue where the promoter of the gene is getting activated. Option 2 is it consists of a promoter or upstream region of a gene along with a reporter gene. Option 3 is we get information about subcellular localization of the protein, and all the statements are true. So the correct answer in this case is option 3. That means option 3 is false because we get information about subcellular localization is when you have a translational fusion construct where your gene of interest and your GFP is being translated into a single protein. So you can see where, using the GFP, where in the cell this protein is localized. But with transcriptional fusion construct, you can only figure out the tissue where the promoter of the gene is active, and it also consists of promoter or upstream region of a gene along with the reporter gene. Okay. So option 3 is incorrect. So according to the question, option C is the correct answer because it is false. Technically, option 4 is also correct, as in uh, it is false. All statements are not true. Since you know option C is not true, this also becomes false. But in this case, you are going to go for the most correct answer, which would be option C. Okay. So option C is the correct answer, or option C is the correct answer. So the question is, which of the following techniques can be used to study protein-protein interaction? Yeast 2 hybrid assay, chip assay, yeast 1 hybrid assay, and RNA in situ hybridization. So the correct answer is yeast 2 hybrid assay. It is going to help you to study protein-protein interaction. Chip assay and yeast 1 hybrid assay is for DNA protein interaction. Yeast 2 hybrid is for protein-protein, and RNA in situ hybridization is for figuring out the RNA in the cell. Okay, so the correct answer is these two hybridization. Cas9 is a exonuclease, endonuclease, peroxidase, and ribosome. So the correct answer in this case it is a endonuclease. It will cut uh, DNA at a certain point within the sequence itself. Exonuclease will cut or degrade the DNA or RNA from the ends. This will cut in between. The endonuclease will cut in between. A peroxidase is an enzyme that will break down hydrogen peroxide, and a ribosome is an RNA molecule that has catalytic activity. Which of the following is an example of targeted suppression of gene expression? Gamma ray and AMS are mutagens. tDNA insertion is for expression of a certain new gene of interest where the tDNA, the borders can be used, the tDNA from agrobacterium, that is, and the borders, the left and right borders, can be used for you to insert your gene of interest into the genome of a plant. RNAi or RNA interference is used for the targeted suppression of a particular mRNA. Okay, and the suppression of that mRNA using RNAi will lead to that protein not being formed. Okay, so you're suppressing the gene expression. In RNA sequencing, dash fragments are of known sequences that are ligated to the synthesized cDNA. So the correct answer is not oligo-DTs, okay, since they are the same. Okay, so they are TTTT, small fragments of TTT. Even if you attach them, you cannot differentiate between different fragments of DNA. Random primers are also not correct because they are single stranded and they attach to complementary sequences. That is not what you want. Adapters are double stranded DNAs that you can attach to your genes of gene fragments, and because the sequence is known of these adapters, and you can use different adapters for the 3' end and the 5' ends, you can figure out, and they can be used to help you sequence the RNA. Okay, and since adapter is the correct answer, none of these is wrong. Okay, and this is the last question for today. That is, in order to discover specific cell markers, the scientists generated a collection of transgenic lines with random insertion of a promoter-less reporter gene and screen for specific reporter activity in the domain of interest. Okay. What is this called? It is gene knockdown is knocking down a gene, so that is not the correct answer. Gene trapping is to figure out if your promoter is active or not. Okay, that is if mRNA is being formed or not. But in this case, you are looking for a specific cell type marker, which is in fact a protein. So this is not the correct answer. Enhancer trapping is to figure out if an enhancer or how much an enhancer is enhancing the expression of a certain gene. So this is not the answer. Promoter trapping is the correct one, where you are adding a promoterless sequence within the exon or the sequence in a DNA that will be in a mRNA and then therefore will become a part of your protein. Okay, so promoter trapping is the correct answer. So that is all the questions for today. I hope that was sort of clear. I'll upload this the video that the recording of the session that I've taken. In case you have any doubts, you can go over that. Apart from that, if you have any doubts, you can ask me right now. If you don't have any doubts, you can leave the meeting. This is the end of the session for today. So I'll wait for a few more minutes to see if there is any doubt that is being asked. Otherwise, you can leave the meeting and I will also end the meeting here. Okay. The two people who are still in the meeting, do you have some doubt? All right, since I don't see any doubts in the chat box, and so I'm assuming there are no doubts, I'm going to end this meeting for all. Okay, thank you so much for joining, and let's meet next week.